The lecture recording is on now. So welcome on this lecture, computers in molecular biology, molecular biology and biotechnology. Today's lecture will be some kind of introduction to the whole topic. Do we have any person which are not Poles? Maybe. Do we have or no? Everybody? No one, yes? So everyone is Polish citizen, so of course I will speak only in English in this lecture, but I just wanted to know if we have any foreigners. So I'm very happy that you are present on my lecture and that we have lots of students here. And in the beginning I will tell you something about computers in molecular biology, taking it from the history point of view, and actually what for and how it can be useful for you as biologist. I hope that this topic will be interesting and maybe some of you will use tools which can be found on the internet, for instance. Very often, most of the tools are actually open, so it's good to know those tools. Any one of you, do you know any tools, any programs which can be used in computers to do something, let's say, in biology? Maybe you can raise your hand. I think that there are some physicists, yes? We have biophysicists here. Let's say people who are in some biophysics departments. I can see some yes or no. Do you have only I suppose that we have students from different topics. Yes, we can have people from, let's say, biotechnology. Yes, do we have who is from biotechnology? Raise your hand, please. It's not funny. You are very shy. Okay, let's go to the lecture. You are very shy, you don't want to tell me that. I will check it on you. I can't maybe possibly check it in USOS, but okay, it's not important. Okay, so let's see a movie which is in the beginning, which is connected to computers in biology, and actually it is some other kind of description which is called computational biology. Yes, so let's try what is computational biology. I will show you a movie.
Computational biology involves the development and application of data analytical and theoretical methods, mathematical modeling and computational simulation techniques to the study of biological, behavioral, and social systems. The field is broadly defined and includes foundations in computer science, applied mathematics, animation, statistics, biochemistry, chemistry, biophysics, molecular biology, genetics, genomics, ecology, evolution, anatomy, neuroscience, and visualization. Computational biology is different from biological computation, which is a subfield of computer science and computer engineering using bioengineering and biology to build computers, but is similar to bioinformatics, which is an interdisciplinary science using computers to store and process biological data. Computational biology, sometimes referred to as bioinformatics, is the science of using biological data to develop algorithms and relations among various biological systems. Prior to the advent of computational biology, biologists were unable to have access to large amounts of data. Researchers were able to develop analytical methods for interpreting biological information, but were unable to share them quickly among colleagues. Bioinformatics began to develop in the early 1970s. It was considered the science of analyzing informatics processes of various biological systems. At this time, research in artificial intelligence was using network models of the human brain in order to generate new algorithms. This use of biological data to develop other fields pushed biological researchers to revisit the idea of using computers to evaluate and compare large data sets. By 1982, information was being shared amongst researchers through the use of punch cards. The amount of data being shared began to grow exponentially by the end of the 1980s. This required the development of new computational methods in order to quickly analyze and interpret relevant information. Since the late 1990s, computational biology has become an important part of developing emerging technologies for the field of biology. The terms computational biology and evolutionary computation have a similar name, but are not to be confused. Unlike computational biology, evolutionary computation is not concerned with modeling and analyzing biological data. It instead creates algorithms based on the ideas of evolution across species. Sometimes referred to as genetic algorithms, the research in this field can be applied to computational biology. While evolutionary computation is not inherently a part of computational biology, computational evolutionary biology is a subfield of it. Computational biology has been used to help sequence the human genome, create accurate models of the human brain, and assist in modeling biological systems.
Okay. Thanks for watching. So I hope that it was quite easy to understand for you. It was quite native speaking computer lady, a bit like artificial intelligence or something. But let's maybe tell something more about history of this whole subject. So you can see here two columns of the slide. On the left, modern biology, and on the right, computer informatics. Yes, two interesting disciplines which started to grow in 50s. So we have on the left side we have modern biology, on the right side we have computers informatics, yes? And there are some interesting maybe discoveries which were important for development of biology. I think that most of you feel to be a biologist, yes? Yes? Please tell me, yes? You are a molecular biologist, somehow. But on the right side, you have computers informatics, which was also developing in those times, in the similar, maybe 50s. So we have here discovery on the left of the DNA, yes, we know this. Sorry? Do you have any questions? If you have questions, I can tell you, of course, I can answer. So I think that you know that those stuff which is on the left about biology, the most important discoveries like DNA of Crick and Watson, and of course we shouldn't forget about Rosalind Franklin, which she was actually forgotten to get Nobel Prize. And of course there was a third guy, Wilkins, which was a Nobel Prize, of course, Nobel Prize winner together with Crick and Watson. Wilkins was actually some kind of collaborator of Franklin, but the story is not very happy for Franklin here. But next, of course, we have some other discoveries, like discoveries of Sander Frederick, British biochemist. So we can see that we have something with amino acid sequences of insulin. So the sequence of insulin was discovered, and he was also a winner of Nobel Prize in 58. I'm not sure if this text is visible there in the back of the, yes? Can you see the text? I will not read the text because I will just talk about it. So it was, I think it's a better idea to have seats closer to the screen. Please come closer to the screen. Come on. Come closer. Come closer. Here. We have a very big room. Okay, students are coming closer. I hope that you have nothing against that I'm recording what I'm talking about. It will be shared to the whole world on internet. So, but it's my creation. Of course, it's a good idea to make some notes also, to make your notes, because you will have something on your notes and you don't. There will be no need to maybe to listen everything once again what I'm talking about, because I think that I should make some easy test, because it is a good motivation to listen. Do we have any PhD students here? I know that PhD students will have some notes, yes, because the rest are without, then they're only just, we will have no 5, 4 or 3 notes. But I would like to prepare some easy test for everyone. It will be not maybe so important to pass this exam, but it will be just some kind of evaluation as you are listening, okay? It will be not very difficult, everything will be on internet, but it's better to make some notes, because it will be easier to just recall something than to listen everything once again. So this short history, not so short because it was from 50s, but my talk. I don't want to tell you very long about this because we started in 50s, but we had to go next into the future to the present time. Yes, so we have many decades to describe. So you can see that in 50s, in the end of 50s, there were also some discoveries about the structure, which were made by Perls and Kendrew. It was about hemoglobin and myoglobin. The proteins, one is in the blood, yes, and the other is in the muscles. This in the blood, the hemoglobin was, I think, I'm not sure, but it was maybe, there's information, hemoglobin X-ray crystallography from human, yes, from human, but the myoglobin was from the whale, the big animal, yes, because there was a need to have lots of protein to make a crystallography of it. Those methods were not so microscopic, let's say, or micromillimeter like today. Today we have a small pipettes, yes, we have small volumes, but those days there was huge volumes of chemicals. So the whale was needed to, or whale meat was needed to get the myoglobin. So on the right, in the same time, we can see some discovery of computers. Yes, the computers which were first based on not on silicon but on lamps. Yes, there were lamps. Okay, you can find some. Oh, actually, before lamps, there were even vacuum tubes. Yes, there were something like this called vacuum tubes, and some unusual. There was the first computer in 1951. How it's called? I have to go to the microphone. Universal automatic computer, yes? UNIVAC 1 system. But next, of course, there was some bulbs. For instance, so next were electronic computers, first electronic computers also in 50s, based on lamps. And of course, first programming languages, like for instance, Fortran. Let's go to the next, maybe decade. So next, 60s. I think 60s was more sophisticated. We can see some discoveries also in bio. And I think the important, let's say, bio discovery which is connected to informatics was discovery of Dayhoff Margaret in 65, American physical chemist, and it was connected to proteins because sequencing of proteins was done in those times. There were no methods to sequence DNA, and she made atlas of protein sequences and structures. You can see on this slide the information about this discovery, about this data. It was actually the first data of proteins, yes, of protein sequences. This data, this base was not very rich in those times, but it was growing in each year. Actually, it was very difficult then to produce such a base because she had to find sequences in different publications. I suppose that you maybe are looking for publications in internet. It's easy. You can type, for instance, something in PubMed and you will get the publication. But in those times, publication were only on paper, yes? So she had to get paper publication when were a sequence, just printed sequence, and she had to put this sequence and to get it together and to put it into computer, yes. And those sequences were, of course, not on hard drive, but there was different types of recording data. So you can see that Atlas of Protein Sequences was published annually as printed books and also on punch cards. Do you know? Do anyone know what was the punch card? The punch card was also mentioned in the movie before. Do you know what was punch card? It was just a piece of paper, actually not maybe paper, soft paper, but like a something which is not soft, and there were just holes. So it was actually the only way of recording data in those days in those computers. Next, of course, were some magnetic types. You can see this discovers this system of recording data on the right. So on the right, you can see some discoveries in informatics, some developments of technology. So, for instance, in 60. There was some PDP-1, Program Data Processor 1, the Computer of Digital Equipment Corporation. And actually, you can see that there were some first even hackers. We can tell that computer hacking starts. So next, in 61, 64. We had integrated circuits. So maybe before there was also in these times was transistor discovery. Maybe there was a bit. Too far. We are too far now. Early 70s. Okay. You can see here in the end of 50s we had some computers which were also based on some newer technologies. Because we had the discovery of transistors, yes. In 56 we have transistor experimental computer 0. It was actually the first transistor computer. The earlier computers were based on vacuum tubes. Vacuum tubes are those lamps, yes? You know, vacuum tube is a lamp which was, for instance, used in old TV sets also. There were black and white TV sets with vacuum tubes or radios. Someone likes, for instance, vacuum tubes for voice equipment because they think that the sound is better. But now actually the vacuum tubes are just a history. So next, transistors, and next, as I said, in 60s, the first integrated circuits. So actually the computers which are more closer to today's computers. And we can see here, we had IBM first server, also some kind of hard drive, let's say. It was a huge thing. It was magnetic because let's say first we had, after the punch cards, we had the magnetic tapes, some kind of big disk storage drive, and next, of course, some in time smaller equipment. And the important thing in informatics is also the Moore's law, that the development of informatics will grow, yes? It will grow not proportionally but exponentially. So those two important things in modern bio and in computers, let's say informatics. But next we have 70s, yes? We have 70s. It's actually time when the computers are smaller, and we have of course some discoveries in modern biology. We have lots of discoveries in 70s. Important things, for instance, restriction enzymes. Yes, you know restriction enzymes as a biologist. We have protein data bank, published in Brookhaven National Laboratory. We have also a first recombinant DNA molecule and a Nobel in chemistry. We have development of DNA sequencing, Sanger Frederick. You know this Sanger method of sequences, I am sure. Yes. Here we have the type of sequences which is based on the gel, but of course it can be also made, for instance, in capillars. Next, 78, recombinant human insulin. And 79, in Dayhoff Atlas, the atlas which was mentioned earlier in 60s, now we have 1081 entries. So 1081 different sequences of proteins. But in the beginning, we had only maybe a few, yes? There were, like here, yes, we have, this is code, but there were not many sequences. And of course, taking computer history, you have faster computers. Because in 17, of course, integrated circuits or microchips are used to produce new computers. Of course, those microchips are very primitive compared to today's, for instance, like in here, yes, or in these gadgets, like smartphones. So we have in 1970 the first edition of Unix operating system. We can see that it is very important system because from Unix, inspiration of the Unix was needed to produce Linux, which is now quite popular in biological calculations. And 73 Unix 4th edition was written in programming language which is called C. This is quite important also, language which can be sometimes useful in biology, but now we have different languages also which can be used. Okay, I will not tell you about the other computers like Xerox Alto. You have this on slides. 74 75, Altair 8800, micro design by instrumentation and telemetry systems. It is this actually wardrobe here. It was as huge as wardrobe. But next, let's say it was the first supercomputer. Yes, it was not a personal computer, but just some kind of those times supercomputer. But next, in 76, Apple I by Jobs, Steven, and Wozniak Steven was made. Yes. So it is the beginning of the history of Apple Corporation. And next, 77, Apple II. This Apple was actually made in garage from parts bought in different shops by those guys. And next, 80s. 80s, it was like years of my childhood actually. So 82, gene bank database was established. You can see that there are lots of things in bio and lots of things in computer informatics, but we have 80s, 90s, 2000, 2002, and next, let's see, the last 8 years was not so very creative, but of course we have still the faster computers, but most of it is now focusing on better smartphones from 10, yes, because I think the 10 is a year when first smartphones were, or some tablets and like that gadgets were on the market, more popular. So we have on the bio on the left, we have gene bank database, we have PCR reaction, yes, you know PCR 83, polymerase chain reaction. We have protein information resource, it's a more sophisticated database. We have FASTA as a program which was used to do first sequence alignments, but it was a beginning of some format which is now commonly used. It was used for DNA and for protein sequences. It was 85. So next, 86. Applied biosystem market, so the first automated DNA sequencing based on, of course, Sanger method. And of course we have also in this year SwissProt database, which now is UniProt. You know UniProt database from different subjects, for instance from some bioinformatic subjects, or from subject which is connected to tools, internet tools used in molecular biology. And we have, of course, the next NCBI 88. It was just before 90s. Yes, the BLAST 19. Do you know those tools very well? I think now. We have human. Of course, those tools are on the bio side, but they are connected with informatics now because they are based on tools, on algorithms. We have 2003 Human Genome Project, yes, which was very important development. It needed very development of many techniques, not only molecular biology techniques, but also bioinformatics techniques. We have the first complete genome of some organism like Haemophilus influenzae, 95. We have Craig Venter Celera Corporation, 98. Competitive commercial project with the whole genome sequencing, yes, shotgun sequencing. We have first NGS platforms in 2004, 454 Life Sciences, a large-scale parallel sequencing. And next, some genome analyzer, 2006, 2008. It's not so far from me. The 1000 Genome Project. 2010, Illumina launched the first high-sec system, the high-sec 2000. I think that some of you knows something about high-sec. Yes, it is, for instance, in our biobank, which is not far from here. And next, of course, some different technologies of sequencing. For instance, in 2017, Oxford Nanopore. I suppose that you know this gadget is quite small. I will not tell more about NGS, but those, of course, all is connected also with bio and with informatics. In the informatics side, you have, of course, some 80s computers like Apple, better Apples, like Macintosh, different brands of computers, like Atari or Commodore and so on, and first IBM PC. And 90, it is just 90 when we have first clones of IBM PC, because IBM just gives this technology to everyone. And this IBM PC, which is the worst computer in those times, actually. It is, most of the personal computers are better than IBM, but the technology is free, so many corporations start to produce different clones of IBM PC computers. So next we have also Linux by Linus Torvalds, some system, free Unix-like system, not for commercial use. And this system actually begins to be very popular in academics, in academic stuff. And of course, we have in 90s we have some GUI systems of this system. So, joining the bio with Linux, we have lots of tools which can be used in computational biology or in bioinformatics, but also in just in some kind of looking for data for biologists. I think our time is going to the end for today. We have 7 p.m. and this will be the end of this lecture. Here we have only in the end some growth of the structures and information. I will tell it later, but please stay a bit, because I would like to tell you something about this Facebook profile, which is connected to the lecture. Actually, not profile, but site, yes, page, internet page. In the end, so you can find on the Facebook, I suppose that most of you have Facebook accounts. you can find the site, the page, which is called like the lecture, like the beginning of the lecture, Computers in Molecular Biology and Biotechnology. You can like it, because it will be useful to find information about the lecture and about today's lecture. I will take the recording to the computer and put it on the slides, and make a video which will be a presentation and the recording of my voice. And I will share it on this site. Yes? So please remember this site and like it. I have lots of likes now, because I was promoting this in you can see. It's about 90, I think. Not so many, no, but 90 people like it, like this, because I was promoting this a bit just before the lecture. But I think that the likes will grow after your participation. And I would like to thank you for your attention, and have a nice evening, and the rest of the, maybe actually have a nice weekend, because it's not far to the weekend. Do widzenia. Do widzenia. Do widzenia. Do widzenia.